Hi, everyone. My name is Michael Muskus, and I'm a segment marketing manager at ATCC. We're so glad you've joined us for the next installment of ATCC's Excellence in Research webinar series entitled High-throughput phenotypic screening to characterize fungal pathogens and monitoring phenotypic drift in bacterial production strains, presented by Dr. Max Cravener, a field application scientist at Biolog, and Dr. Shaheen Ali, a senior scientist at ATCC. In today's presentation, Dr. Cravener and Dr. Ali will describe the research partnership between ATCC and Biolog to leverage advanced phenotypic screening to characterize fungal pathogens and monitor phenotypic drift in bacterial strains. This innovative approach is crucial to ensuring the reliability of bioproduction and experimental results. During or after the webinar, if you have any questions for our two speakers, please use the chat function available through the webinar program. All questions will be answered as allowed at the end of the presentation. The recorded presentation will be archived on the ATCC webinar website at www.atcc.org. But first, a little about ATCC before we get started. ATCC is a nonprofit organization founded in 1925. We are headquartered in Manassas, Virginia, with additional facilities in Gaithersburg, Maryland. ATCC is a leading global supplier of high biological materials and an ardent collaborator with the scientific community, which leads us to today's talk. So with that, I'd like to welcome Dr. Max Cravener and Dr. Shaheen Ali.
Thanks, Michael, for the kind introduction. So, as Michael already explained about a little bit about ATCC, and he told that ATCC and Biolog, we started this joint project last year. Before we go into the actual project, I would like to give you a little recap about ATCC's microbial collection. So, our microbial collection is a huge collection. So we have more than 70,000 bacteria, fungi, virus, and protozoa in our collection. We have more than 1,300 different microbial type strains. And our bacterial collection include almost more than 1,200 different genera. And then we have more than 1,800 different genera of fungi. And we have virus from almost around 200 different genera. So many of these microbial strains are part of standard and guidelines for various organizations like USDA, USP, ISO, FDA, and around 475 strains are recognized as reference material for quality control. So ATCC also, beside the live organism, ATCC also provide inactivated material and nucleic acid, and we use various advanced technique to characterize and authenticate this material. Now, beside providing these microbes and standard to the greater community, to the research community, we have also another mission, a mission to accept, authenticate, characterize, and store all the new and novel microbes. And the importance for that mission is, I'll give you a classic example, which is Zika, because Zika was first identified in 1947. And it was deposited in that year to ATCC collection. And that virus was in our collection for more than 50 years. Nobody wanted it. And then finally, when the major Zika outbreak occurred around 2013, everyone wanted the virus. And so basically, we distributed this virus around the world, helping different researchers and regulatory bodies like in developing identification kits and various therapeutics. So this is the importance of this kind of collection. But today we are not going to talk about our whole collection. Today we are going to mainly focus on two topics. One is the human fungal pathogens, and another is the quality control issue that we face in this kind of microbial strains. So regarding the fungal human pathogen, which is becoming a very like a big problem in recent years. And in US alone, the economic burden for fungal disease is almost more than like $11 billion. And that's why WHO has recently released, in 2022, they have released the first fungal priority pathogen list. And when that list was released, we went into our collection and we identified all the strains from these species reported by WHO. And you can go to our ATCC website and read this blog about WHO-related priority fungal pathogen list. So today, Max is going to talk about that how the new Odin platform, Biolog's Odin platform, can characterize some of these fungal pathogen and why it is important. And nextly, today we're also going to talk about the traceability and reproducibility crisis. Now, this is a very hypothetical situation, like where you can see that someone deposit a microbes to ATCC and then some researcher buy it from the ATCC collection. But then this is very common phenomena in academia where you get a microbes from a colleague or someone down the hall in the lab. So basically, people like passes those microbes, then give it to different labs. It goes to different labs, and finally, they sequence and submit in like NCBI. So it is very common to have multiple sequence of the same ATCC strain in NCBI collection. So that's why we looked into the NCBI collection and we identified some of the ATCC material, like say this is Acinetobacter baumannii 17978, where we have 7 different sequences available in NCBI, or say this Candida albicans has 2 different sequences. So what we did, like we sequenced our ATCC material in-house. We did a very in-depth, high-quality sequencing, and then we compared this NCBI sequence with our sequence. And not to be our surprise that we found a few SNPs, but in some cases you can see like there are hundreds of thousands of SNPs. So this is a huge problem. So basically it shows that when a microbes roll over from lab to lab or within the lab, it's cultured multiple times, they acquire all those genetic mutation. And we don't know how the phenotypic variation will be there. So that's why next in this today's seminar, Max gonna focus mainly on how the phenotypic changes occur, like if we passes these microbes a few times, like 20 or 40 times. So with this, I'll hand over to Max. He's gonna discuss more about the research and the findings that we have.
Thank you, Shaheen, for the introduction and all the background information. And so, as he mentioned, I'm going to now talk about the phenotypic identification method that we use here at Biolog, as well as the characterization pipeline for these WHO pathogens from the priority list. So the WHO fungal priority pathogens list serves to inform clinicians, researchers, government officials, et cetera, on these pathogens of interest and why they're important, give them a lot of information about how critical they are to study, what they can do in the public health setting, and then any unmet research and development needs that may be out there regarding these strains. And they're divided up into 3 major groups: the critical, high priority, and medium priority groups. And you can see that the critical priority group, thankfully, is pretty small compared to the other 2. And so in order to characterize and add these to our identification database, we chose 11 different species from the priority list, including all 4 from the critical priority group: Cryptococcus neoformans, Candida auris, Candida albicans, and Aspergillus fumigatus. As somebody who's worked in the fungal field for several years, these are the poster children for fungal pathogen research, and so we wanted to highlight those. And then from the high priority group, we have Candida glabrata, Tropicalis, and Parapsilosis. Again, some important things to study. And then from the medium priority group, we have another 4, Scedosporium and prolificans, Tularomyces marneffii, Candida krusei, and Cryptococcus gattii. And so for each of these, we chose two isolates of each species. That way we could capture some more variability and not just tie our identity profile to one strain. So the way that our identity method works here at Biolog is using a microplate-based method. And so we have two plates that are used for fungi, our sporulating fungal plate or filamentous fungal plate, and then the yeast identification plate. So the way that these work is that each well here has a different substrate in it, mostly metabolic substrates and some resistance ones as well. And then we have a redox dye included in there that acts as a reporter for metabolic activity. And so cells are collected directly from the solid growth media, whether that's spores or conidia for the filamentous fungi or just yeast. And then those are resuspended in an inoculation media and then inoculated at 100 microliters per well across the YT or FF microplate, and then incubated, and we can run the ID analysis from there. And so this technique is really nice because it doesn't include any DNA extraction or any other higher level steps to get an identity. All you have to do is pick colony and inoculate a microplate. And then these are incubated and read kinetically in Odin for up to 96 hours every 20 minutes. In each read, it'll attempt to make an identity, and most of them identify within 24 hours or even sooner. But for the sake of collecting data for these new profiles, we incubate for 96 hours to really capture that entire profile. A little bit more about the Biolog identity ecosystem. So we have a couple of different instruments that are able to do this. Here we're going to be showcasing the Odin instruments, which are our kinetic identification and characterization platform. But we also have our ID station, which allows for endpoint identification. And so all of these systems use this single color chemistry that can be used to measure metabolism of any nutrient substrate that are in these wells. And so the analysis of the color development at either OD490 for filamentous fungi or OD590 for YT, and then turbidity gives us an accurate identification profile to the species or even strain level in 1 to 3 days. So the Odin database contains over 2,900 unique taxa, which are comprised of more than 1,500 aerobes, 360 anaerobes, 260 yeast, and 700 filamentous fungi. And this number is growing all the time. So the workflow here for adding these new pathogens to our database, and this is something that you could do yourself if you wanted to add your own things to a custom ID database. We took these 11 fungal pathogens, a couple strains per pathogen, ran them on 5 YT or FF microplates, depending on which one was more relevant. And this is to capture some of that day-to-day variation. We don't just want to be making an identity profile off of one plate of data. And then once we have those plates of data, we then compile it and test that profile against everything else in our database. This makes sure that, one, the profile in itself is consistent, and two, that we're able to differentiate it from everything else in our database. And once that's done, we then add them in, and then we can push out that update to all of our users, and which we've done at this point for these strains. So I wanted to talk a little bit about Candida auris. So Candida auris is the one that we use 4 strains for instead of 2. And the reason for that is because it's a really important pathogen. It's in that critical priority group. Candida auris is naturally multi-drug resistant, which has caused a lot of issues both in the clinic and in research. And so it's really important to be able to know not only if you have Candida auris, but also if you are able to identify which strain of Candida auris that you have, because different strains can have different phenotypes. But this is really difficult to do with traditional methods like sequencing or MALDI-TOF because the genome sequences and the surface proteins are very similar from one strain to another. And so our hypothesis was that we were able to differentiate these with our YT identification plate, and lo and behold, we were able to. And so what you're seeing here is the absorbance area under the curve for each of the 4 clades representatives, and we can see that they have very unique absorbance profiles regardless of what strain they are. And so, for example, here at the bottom, this MYA 5001 isolate, which is clade B, was the only one able to utilize these carbon sources here as the sole exogenous source of carbon. And so using those differences, we're able to actually differentiate these to the strain level, which is something that is very difficult, if not impossible, to do with sequencing in Molotov. So now that we've added these strains to our identification database, we now wanted to establish a more broad-reaching phenotypic profile for the pathogens. And the way that we wanted to do this was using Biolog's phenotype microarray set of microplates. And so PM, or phenotype microarray plates, is a set of plates. There's 25 of them that are tuned for either bacteria or fungi. And these probe everything from metabolic substrate utilization to pH and osmotic stress resistance, or antifungal and antibiotic resistance as well. So if you use dye and no dye side by side on the full panel of PM plates, you can test your organism in up to 4,800 unique tests in a high throughput fashion using Odin. And each of the substrates are designed and chosen in order to probe specific pathways and to give you functional information that you can act on after running a PM plate. So they're not just random carbon substrates or other things. It's specifically chosen in order to allow you to probe very specific points of the important metabolic pathways in your organisms. So using the Odin system, we're able to kinetically read and incubate and analyze all of these plates at the same time. So Odin L is able to handle 50 plates at a time, or Odin 8 is 8 plates. So here we're using Odin L and being able to run all 20 plates side by side for multiple organisms at once. And doing so, we're able to collect a few different parameters, or calculate a few different parameters surrounding that raw data, including the average and maximum curve height, the area under the curve, and then the average and maximum metabolic or growth rate. And so these parameters allow you to make a lot of conclusions about your organism. And additionally, when you plot the curves of your different samples next to each other, you can also see the lag times in how long it takes them to start growing or respiring in the presence of a given substrate, which can also be very important information, knowing how long it takes your organism to adapt to a given situation. So the setup for this is relatively straightforward. We ran PM plates 1 through 10, which are generic for any microorganism, and then PM 21 through 25, which are our antifungal resistance plates. And we chose to measure growth here, so that means that there was no dye used. We're just doing growth curves. And we read the plates at OD 590 every 20 minutes over the course of 72 to 96 hours, depending on the organism. And so for the filamentous ones, those are run at 26 degrees for 96 hours. And then for the yeast, those are at 30 degrees for 72 hours. And the plates that we're testing on here, just to give you an overview of what they all cover, PM1 and 2 are carbon source utilization assays, where the carbon source in the well is the only source of carbon they have. PM3, 6, 7, and 8 are nitrogen utilization assays. PM4 is phosphorus and sulfur. 5 is other nutrients. And then 9 and 10 are osmotic pH stress. And like I said, 21 and up are the chemical sensitivity assays. So some broad summary of the results before I take a deep dive into a few examples. The growth that we measured for each of the 6 species on at least one plate, if not replicated, we found numerous different substrate differences. So different organisms preferred different carbon sources or nitrogen sources, or took longer or shorter in order to start growing in the presence of those sources. And they also all had unique susceptibility profiles to various drugs and stress conditions as well. To give you a few specific examples, first up, Aspergillus nigulans, stress resistance. So here in PM9, our salt stress plate, we can see that as salt concentration increases, as going to the right, growth falls off quite precipitously, even at the middle to lower concentrations of salt. And similar story here for urea. Increasing concentration seems to knock down growth quite significantly, as well as sodium benzoate. An acidic pH. And then here with PM25, this is a drug resistance plate. And so we can see that Nigilans has varying resistances to a couple of different antifungals on here, or different inhibitory compounds. And so I pulled out tobramycin and malic acid, and just to show that it has resistance to tobramycin at all concentrations, and then malic acid is varied depending on the concentration. Scedosporium prolificans, another one of the filamentous fungi. Here we have PM10, which is the pH resistance plate. And so rows E through G are all at pH 9.5 and using different buffers. And so we can see here that basic pH is really challenging for S. prolificans and delays growth. While it is able to grow in these alkaline conditions, it does take a while for it to adapt to that condition. And if we look over here at PM22, this is another drug resistance plate, has, again, varying resistances to different compounds, including, for example, this sodium metavanadate is very resistant to that. And for CCCP, the resistance falls off as you go to higher concentrations. Tularomyces marneffii, another filamentous one. So we're looking at a couple of stress plates here. So PM21 and 24 antifungal plates. And again, these varying amounts of resistance to each of the drugs that we chose, or each of the drugs that we screened it against. Now into some yeast. So Candida albicans is endemic on almost every human on the planet. So it's a very important organism to study, again, in that critical group. And near and dear to my heart. It's something I studied for a long time. And we see it has its own unique resistance profiles to different antifungals. And for example, down here, fluconazole, this is our first line of defense, usually over-the-counter kind of drug for Candida infections. And this strain in particular was susceptible to any concentration of fluconazole that was used on the plate. And now into Candida auris. Here I have PM1, which is our carbon source utilization plate, or one of them. And alpha D glucose here acts as sort of a positive control. And we can see that it has a nice and robust growth curve here. And then comparatively, fructose, mannose, and maltotriose also had similar growth curves, indicating that it likes those carbon sources as a substrate. And then over here, PM4, this is our phosphorus and sulfur utilization plate. And this showed that it was able to take up almost all exogenous sources of phosphorus and sulfur that we gave it, and was able to utilize those as its sole source. Looking at some stress resistance for auris, on the left here, we have PM9, which is the salt resistance plate. And again, slightly decreasing at the very highest concentrations of salt. This is around 10% salt. We finally are seeing a reduction in growth, but it's able to grow in the presence of even almost the highest concentrations here, in contrast to the Aspergillus nigilans that I showed you at the beginning, which fell off really fast. But it also shares that susceptibility to sodium benzoate as well. Over here on an example of a drug resistance plate, PM22, we can see that Candida auris is in fact resistant to almost every drug on the plate. And this was reflected on the other drug resistance plates as well. But I wanted to pull out here triclosan, which is a common household disinfectant found in hand soap oftentimes. It was very susceptible to that. And it's also susceptible to thallium acetate on this plate as well. So overall, the take-home message I want you to get here from these phenotypic profiles is that it's really important to generate this baseline, this comprehensive profile for any important and emerging pathogens or strains that you need to work with in your lab. And using Odin to do this can allow you to do it not only quickly, but also have the power of the analysis software as well to calculate a myriad of different parameters. And again, with this system, you're able to not only characterize these strains, but also identify beyond the species level where other methods like sequencing and MALDI-TOF may struggle. Moving on to the second half of the talk in monitoring phenotypic drift in bacterial production strains with Odin, I'm going to talk about that reproducibility crisis that Shaheen mentioned. So monitoring drift in general is a challenge. And so we know that mutations happen over time. And so genetic monitoring is an important step that we do. As Shaheen mentioned, you can have those scenarios where a lab submits a strain to ATCC, and then decades later, a scientist buys that stock, and then they transfer it to more and more labs, and then 20 years goes by, and then another lab down the line resubmits the sample to ATCC, but now it's drifted genetically and phenotypically, and may not be as similar as we would hope. Unfortunately, monitoring is expensive and prohibitive depending on how you do it. So genetic monitoring requires full genome sequencing, which is very training intensive and requires a lot of special knowledge and also a lot of prep and time just to get the results back. And then phenotypic monitoring also can be really prohibitive because dozens to hundreds of different phenotypes need to be assessed because organisms don't just do one thing. And the Biolog phenotype microarrays, along with Odin, allow you to not only rapidly test your phenotypes, but also get an early warning as to whether or not your strains are drifting. And using the system, you can probe, as I've mentioned before, describing the phenotype microarrays over 2,000 unique conditions, just if you use dye, or if you use no dye and dye side by side, measuring growth and metabolism, that number goes up to over 4,000 different conditions and tests all in one. So it's really important to monitor both genomic drift and phenomic drift. And I hope that through the next few slides, I convince you of that. What I want to drive home is the fact that the link between the genetic changes and the phenotypic changes might not be as straightforward as you would think. And that's because genetic changes could be in a wobble base, it could be an amino acid that's in an unstructured domain that doesn't really have an impact on protein function, or it could even be in a non-coding region and therefore not informative. And phenotypic changes can be impacted by epigenetics, methylation patterns, and other different changes that can go on that can alter how proteins function without actually changing their sequences. And the overlap between these is often very difficult to identify. So it's important to monitor for both. So again, we know the genetic drift occurs over time. Mutations accumulate. This is a known thing in biology. However, it's unclear how often these translate to phenotypic drift as the strains are continuously passaged. So the hypothesis that we wanted to test was if bacterial strains are passaged many times, then we will be able to detect the phenotypic variation relative to the original strain using Odin-L and phenotype microarrays. And additionally, we'll be monitoring the genotypic drift in the background as well. And the experiment we designed for this was to passage E. coli, Streptococcus thermophilus, and Lactobacillus casei, fresh stocks from ATCC. We would passage those every 24 hours and take samples at defined passages and screen those for drifted phenotypes and sequence their genomes. The experimental setup here was to incubate them on rich media. We don't want to induce any selective pressure here. We're just looking for natural mutations and trying to simulate the passaging that one might experience giving a strain from your frozen stock to your lab mate's frozen stock. And so for E. coli, we used our Biolog universal growth media with 5% blood, 36 degrees every 24 hours to pick a colony and subculture. S. thermophilus, we used M17 supplemented with lactose, incubated with CO2. Passage every 24 hours, and then L. casei was medium 416 and incubated again with 5% CO2. This time it was passage every 48 hours due to its slower growth rate. So E. coli results. Passaging E. coli, we looked at passage 0, the reference strain, and passage 20 on all 20 PM plates, and then found any significant phenotypes, and then looked at those further with passage 40. And so this was done on Odin for 24 hours at 36 degrees. Reads were taken every 20 minutes to get that kinetic curve. And we actually identified over here a few dozen different phenotypes on several different PM plates, including one nutritional plate, PM3, and then several stress plates. And then when we repeated the assay on P40, after 40 passages, we again found more phenotypes that came up. And so these are divided into a couple of different categories. So we have our metabolic phenotypes and the sensitivity phenotypes, and then we have those that appeared in P20 and then got worse in P40, and then those that appeared in P20 and stayed the same, or those that just appeared in passage 40. And you can see that by passage 40, we're gathering quite a significant number of mutations or phenotype changes as passages go on. So to give you an example of the metabolic phenotypes here, PM3, this is our one of our nitrogen utilization plates, has a variety of amino acids and amines on it. And if we overlay P20 versus P0 and then P40 versus P20, we can see that anywhere there's pink, that indicates that the passaged strain was worse at using that substrate than the older strain. And so here we have only a few are called out as meeting the threshold by passage 20. However, by passage 40, that number goes up a lot more, where we have almost a dozen different phenotypes, passage 40 versus passage 20. And so this ability to take up and use these nitrogen sources is obviously getting worse. And so we ran some statistics on this. This figure is looking at passage 40 versus passage 0, and ran a statistical comparison test and found that these are in fact significant differences in the ability to utilize and take up these nitrogen sources by passage 40. To drill down a little bit into the resistance phenotypes that we saw for E. coli, again, we have a few different phenotypes that either appeared in passage 20 and got worse, like this chlorotetracycline resistance, where in passage 40, it starts to drop off more in the lower concentrations here. And then we have others like this fusaric acid resistance phenotype, where only after 40 passages has something changed and the resistance is going down. And we see similarly, this 2-nitroimidazole and sodium cyanate also have increasing susceptibility. And then cefamidazole is one of the few drugs that actually it gained resistance to, which is unusual given that we are not exerting any selective pressure here. So this is just a natural gain in resistance. So again, like I mentioned, we also did whole genome sequencing on these strains to identify any SNPs or variants that may have appeared. And doing so, we found that there were 10 different variants by P20 with an additional 5 that appeared in P40. However, most of these did not have a very high frequency. You can see here, these are our top 3 variants that are in coding regions. Some of the other higher ones were in non-coding regions. But these only had up to 60% frequency by the time passage 40 rolls around. So we know it's a mixed population. And the genes that are being affected here are pretty varied. So we have part of the type 6 secretion system has been mutated, a galactoside acetyltransferase, and then a ribosomal RNA. And the rest of the mutations were in non-coding regions. So some conclusions here, the phenotypic drift occurs more rapidly than expected. When I was doing the literature review, developing this project, I was finding things that said, 50 to 100 passages might be necessary in order to find anything, but we were able to identify these phenotypes in as little as 20 generations. And so while some of them are more stable, others get worse over time, meaning that pathway is more susceptible to changes than others are. So Streptococcus thermophilus was the next one. This is a common production strain. So again, we used passage 20 and 40 strains on PM 1 through 20 and looked for significant phenotypes here. And so we found those on pretty much all of the resistance plates, so PM 9, 10, and then 14 through 20, and found 14 different sensitivity phenotypes that had changed by P20, and then an additional 12 that popped up by P40. Strep thermophilus did not appear to exacerbate phenotypes. It seemed more to collect them instead. And typically, there were no significant changes that were identified in metabolic phenotypes in either P20 or P40, which was kind of surprising that it was all resistance phenotypes here. And so a couple of examples, this is an iodo-nitrotetrazolium, increased resistance by P40. And then over here in PM9, this is our salt resistance plate. We can see that at different concentrations or different types of salts, it was starting to feel that stress and reduce its growth rates in those substrates. To give you a zoomed in view of some of these as well, again, INT had increased resistance, surprisingly, in P20 and P40. It was pretty stable. And then similarly for thiosalicylate had decreased resistance, again, stable after 40 generations, and similar story for these other inhibitors as well. And so by P40, a lot of these phenotypes were seeming to be related to cation stress. And so, for example, INT resistance, increased INT resistance indicates a shift away from respiration because INT pushes the organism towards non-respirative growth. And so that was interesting as we were passaging Thermophilus in an aerobic physiological CO2 environment, that it is shifting away from respirative growth. So we also did genome sequencing here as well. And we had a lot of different mutations that we were able to identify in Strep Thermophilus relative to the P0 reference. There's actually 22 different mutations, or 26 actually, sorry, different mutations that had over 99% frequency by P40. You can see here that only 2 of them had over 99% frequency in P20. However, all of these now were up to being the dominant variant by passage 40. And so this is these are a bunch of different kinds of genes here, and I'm not even showing the non-coding regions or the theoretical proteins. So we have some ABC transporters, ribosomal RNAs, some undefined domains, a CRISPR-associated protein, some transporters, et cetera. Any of these could be related to some phenotypes that could pop up in the future, or they might not be impactful at the moment. And it's very difficult to tell, which is why we do the phenotypic monitoring. Lactobacillus casei was the next one. So this is a common strain in the dairy industry. So here we had a little bit of trouble getting it to grow consistently, and it stopped at P15 because it was no longer growing in non-anaerobic conditions. And so rather than switch conditions, we just screened the P15 generation, again on PEM 1 through 20. And we actually found 8 different sensitivities that had changed over time. And a couple of examples here, thioctic acid resistance went up and chlorambucil resistance went down. And so this is really, again, surprising. Like I mentioned, we were shocked that we got phenotypes as early as 20 generations, let alone 15. And so this really drives home the point that we need to be monitoring how often we're passaging strains. And if we are passaging them, to make sure that we run them on a phenotypic panel or every so often to track them. And again, even in just 15 generations, we picked up a few 100% frequency mutations, including 7 SNPs and a deletion compared to P0. Most of them were in non-coding regions, but these 5 here were in coding regions. Again, hard to match to the phenotypes, but that just, again, emphasizes the need to do both phenotypic and genotypic monitoring. So both of these monitoring techniques revealed unique changes in all 3 of the production strains that we looked at. And there are only a few of them that really were able to even conceptually link to each other. Without doing further mutational studies, again, it's really hard to assign causality to various genetic changes. And so just being able to do the phenotype screen, we're able to find dozens and dozens of altered phenotypes relative to the unpassaged variants of these strains. So again, just the importance of monitoring both genetic drift and phenotypic drift. I can't understate that enough. So we know that these strains are passaged. This is part of lab life. It is part of how microbiology labs have functioned for decades. And it's not going to stop. This is just a fact of life for microbiology research, but we can take steps to limit the effects and to monitor our strains and make sure that they're performing consistently and that we know what is happening and know as much about our current isolate as we possibly can and not just fall back on reference data. and monitoring for both phenotype and genotype, whether it's on WHO-listed fungal pathogens or high-value production strains. Having that solid foundation of phenotypic profile to supplement or complement your genotypic profile is really critical. And with that, I will pass it back over to Shaheen to round out the talk.
Thanks, Max. So as Max explained everything, so the final take-home message is that you should establish a baseline for this important strain. And the Biolog phenotypic microarray Odin platform can streamline this screening by screening more than 2,000 unique conditions simultaneously. And ATCC microbial stocks will provide a reliable, authenticated material with traceability. Now, this is in a nutshell how our ATCC system works. So basically, when we receive something from a depositor, it goes through QC, which involves growth and observation, then sequencing of 1 or 2 genes like ITS or 16S, and then we do the whole genome NGS sequencing. And once we are satisfied with the strains, then we produce safety stock, seed stock, and distribution stock. And whenever we need to produce distribution stock, we go back to the seed stock. So this is how we minimize the passage. So either you should have a similar model in your lab or in your research institute, or you should get authenticated strains from ATCC or similar repository. So with that, I'd like to thank you, and we are open to questions.
Thank you, Dr. Cravener and Dr. Ali, for the excellent presentation. In just a few moments, everyone, we will begin our Q&A session. Please use the chat function available in the webinar program to submit your questions. And remember, the webinar presentation will be available on demand at www.atcc.org and also on the Biolog website at www.biolog.com. Looks like we've had several questions come in, so let's go ahead and start with the first one. Max, or Dr. Cravener and Dr. Ali, how many cells colonies are required to run a full set of PM plates?
That's a great question. So for a full set of PM plates, it really does not require that many cells. So usually to inoculate a full set of 20 PM plates with bacteria, you're looking at around 10 colonies worth of cells. So it's pretty minimal as far as what you need. And for fungi, that's usually, you know, one colony that's about the size of a dime that's sporulating. That gives enough canidia or spores in order to run an assay.
All right, sounds good. The next question we have says, how does Odin characterize anaerobes without being in an anaerobic chamber?
Again, the nice question. So the way that Odin works with anaerobes is that we inoculate the plate in an anaerobe chamber or other anaerobic environment, and then the plate can be sealed. We have a recommended plate seal we use for this, and then that can be incubated and read kinetically in Odin. Even though Odin is out in the aerobic atmosphere, we're still able to preserve that anaerobic environment within the wells for up to 48 to 72 hours. Depending on the plate seal that you use. And this is exactly what I did with the LKC isolates in this study.
Okay. We have another question. Dr. Cravener, I think this one is for you. Drug resistance changed in many cases in the production strains. Do you think this would be different if they were passaged with drug selection?
Yes, I do think that it would be different. So we were particular about using non-selective conditions in this study in order to replicate the passaging that would occur, you know, just on the bench, giving strains to fellow lab members, et cetera. But I think that we would probably see less of the sensitivity phenotypes change, or perhaps different sensitivity phenotypes would change whenever there is drug selection during passaging.
Okay. The next question, how did you determine which parameter to use to compare strains? And a follow-up, how did you determine the cutoff for biological significance?
Yeah, so with Odin, we're able to calculate a few different parameters, looking at curve height or rate of dye reduction or growth or area under the curve, and all these are informative in different ways. And so for this study, we chose to use area under the curve, as this is a general overall phenotypic output or overall growth over the time of the assay, and this is more of the general conclusion parameter that I like to use a lot for these types of studies. Maximum rate and maximum curve height are also informative, but sometimes for comparison over a long assay, it's really beneficial to use area under the curve. And then for the cutoff, that is determined by using the background wells, the negative controls. And so typically what I do is look at the two strains and then what the difference is between those in the negative control, and then use that value multiplied by 2 or 3, and then that is the baseline cutoff. Of course, that can be tuned depending on your specific assay, but in this case, we used a 2x fold cutoff.
Okay. Let's see, we have another question that just came in. Can mixed colonies be identified by Odin? So Dr. Cravener, this would be for you.
It's a great question. So Odin only works for identification for pure cultures. You will need to streak for single colonies and purify those before running those plates on Odin, yes.
Okay. And another question from a different submitter. How many replicates do you have on a plate?
So replicates on a plate, that's a great question. So for the PM plates, those are single determinations, so every well is going to be unique. However, we do have some plates at Biolog that have internal replicates, such as our eco plate and prebiome plate, but the PM plates are all single determinations, yeah.
All right, good question. Let's see, we have another one that looks like it just came in. Do I have to use Candida spores for filamentous fungal ID in PM?
Yeah, so, yeah. So when it comes to the cell type that you're using for PM assays, it's really flexible on what you're using. So I've used mycelia or hyphae as well as canidia and spores. So really the biggest thing to consider is what metabolic state that they're in, and so trying to synchronize everything so that the wells all are growing at the start growing at the same time is usually why we lean towards canidia or spores, because those will typically germinate around the same time, not always. But for identification purposes, we do require that you use canidia or spores when available, although some filamentous organisms don't produce those, we understand. But generally speaking, yeah, the canidia or spores are the best way to go because they're in the same metabolic state starting out.
Okay. Sounds like that was very comprehensive. Here's a question probably for either one of you. How are samples at ATC stored long-term? And if it is using LN2, is it in the liquid or vapor phase? And how do you control for cross-contamination?
Yeah, hi. So normally in ATCC, we store them in liquid nitrogen in vapor phase, the frozen stock. But we also produce lyophilized stock. So basically those are distribution because it's easy to distribute lyophilization stock. You don't need dry ice. Those are for distribution. But all the seed stock or for long term, we use like glycerol stock and store at liquid nitrogen vapor phase. And normally, yeah, another like how we like minimize the cross contamination because it's normally it's inside the tube, and we use separate box or separate samples. So, and under vapor phase, you get less cross contamination, almost zero. Yeah.
Okay, thank you, Dr. Ali. Another question that just came in, how do you store the samples?
Yeah, so I'm going to, go ahead, Shaheen.
Yeah, it's the same question. I think I just answered. So basically, we store them in liquid and paper. Yeah.
Yeah, and for the PM study here for passaging, we stored those in glycerol stocks.
Okay. Another question that just came in. What is the reference method used for authenticating the ID by Odin?
Yeah, so the reference method that we use, so we have a database of existing ID profiles that we'll compare against to make sure that the profile is unique when we generate one. But when we are faced with an unknown strain, a true unknown, and you run it on an ID plate on Odin, you'll get a no ID result. And then you would be prompted to identify using another method. And so at Biolog, we usually use sequencing or MALDI-TOF in order to do that. Once we get a confirmed ID, then we can add it to the Odin database to get that more rapid and easy identification workflow.
All right. Next question. Do you read the plates with the lids on or remove the lids?
Lids on. Yeah, Odin reads from bottom to top and reads right through the lids.
All right. Another question, how do you determine if changes you detect between strains are within error?
Yeah, so it's a great question. So the software will do the pairwise comparisons by itself, just subtracting tests from reference. But then I also do some offline quality control, looking at whether or not the signal is above background. And so I'll go through and take the geomean of the negative control wells and use that as a background control and subtract that out from everything, and to make sure that my that the biologically significant phenotypes are in fact above reference. Yeah.
Great. Okay, this next question, Max and Shaheen, thank you for the nice presentation. How do you determine which concentrations to test? And do you make a determination of which level of resistance is relevant to the environment?
Yeah, so our plates, the resistance plates, are formulated to use industrial and clinically relevant concentrations of drugs. So these are comparable to the ranges you would see in MIC testing. And those are all based on, those are all to be generally applicable to as many microbes as possible. And so each drug concentration is going to be slightly different based on what works best according to the literature.
Great. Another question that just came through. What level of variation is reflected in MALDI-TOF? Presumably, it's much lower than Biolog. I'm not sure if that's clear. What level of variation is it?
Yeah, so MALDI-TOF can be very tricky to use sometimes, mostly because of the sample prep, but also because surface proteins are sometimes really similar depending on the strain that you're using. And so I'd like to go back to that Candida auris example that we have in this presentation, where using MALDI-TOF and sequencing, it's really difficult sometimes to parse those isolates or those different strains from each other. Whereas with the Biolog phenotype ID plates, we are able to differentiate them based on their carbon source utilization and ability to grow in the presence of different carbon sources. So we would have a lower error rate, I think, based on MALDI-TOF, just in the fact that we're able to use more tests and have a wider breadth of substrate utilizations that we can use to call an ID for very closely related strains.
All right, thank you. Here's a great question. Is there a possibility for research collaboration? So how do people maybe get in touch with you? And yeah, so I think there's some interest here for probably both of you.
Yeah, ATCC is always open to research collaboration. Yeah.
Yeah, same with Biolog. You can email that sales at info@biolog.com and just mention that you're interested in collaborating, and then that will get transferred to the most relevant person at Biolog.
Yeah, same way. I think you can email in that ATCC email, so they will direct to the actual scientist. Yeah.
Great. All right, here's another question that just came through. This is great, getting a lot of questions here. How about the ID station? Do you remove the lids to read the plates? You may have mentioned this earlier.
So we haven't talked about ID station at all in this talk, but it does the same thing as Odin, so you can read through the lids, so you no need to remove them.
All right. And that concludes, let's see, just let me check. Yep, that concludes the Q&A session, everybody. I want to thank everyone for attending this webinar. This concludes the webinar as of this time. Also want to give thanks to Dr. Craver and Dr. Ali for the time in preparing the webinar and presenting and answering all the questions today. For the attendees, if you'd like to learn more about Biolog, including collaborations, as Dr. Cravener said, you can email them at info@biolog.com. You can also visit their website at www.biolog.com. And if you'd like to reach out to Dr. Ali, feel free to email him at sales@atcc.org. And to find out more information about ATCC, visit our website at www.atcc.org. Also, wanted to say, please be sure to sign up for our monthly newsletter, plan to attend our next webinar, and listen to our podcast called Behind the Biology by Brian Shapiro. You can find this information at www.atcc.org/resources. I'd like to thank everyone again for joining the webinar and have an amazing day. Thank you.